What's up?
Antigen presentation is a process when our immune system determines if substance is antigen or it's not antigen. We have two types of antigen presentation. The most common type occurs by MHC II receptors when substance is presented to T helpers. In this process, we have substance, antigen presenting cell with MHC II receptor and T helper. Also, we have the second type of antigen presentation that occurs by MHC I receptor when substance is presented to T killers. Let's take as an example the most common antigen presentation with MHC II receptors. MHC II receptor is a protein receptor on the surface of antigen presenting cell. The most important feature is that MHC II receptor is a protein, not gene, and the function of this receptor is to present substance to T helper. But the genetic information how to make MHC II receptor called HLA gene, which means that it's nucleotides in the DNA molecule. And this HLA gene is located on chromosome 6. So HLA is basically an instruction how to make MHC II receptor. When antigen is presented to T helpers, it's T helpers that make a final decision. If they decide that substance is normal, there will be no immune response. But if only they would think that substance is something abnormal, they would start immune reaction against this substance. Why HLA genes are important? Because some HLA genes can be genetic risk factors for autoimmune disorders, as celiac disease, lupus, or rheumatoid arthritis. So we can use HLA genes as a screening method that would help us to make a diagnosis. And to understand how it works, we have to know how antigen presentation occurs.
I got an idea. This might be a little crazy. Crazy's better than nothing.
So let's suppose that this guy is the substance and we want to determine is this guy dangerous or not. And there is nothing wrong with this guy, so let's suppose that it's our friend. Initially, macrophage intake substance in order to present this substance to T helpers. And antigen presentation occurs by an MHC II receptor. What we have to know about MHC II receptor is that it's a protein. And the genetic information that tells us how to make MHC II receptor is contained in the gene that is located on chromosome 6. And we call this gene HLA-DRB gene. But we have different variations of HLA-DRB genes, and such variations we call alleles. The simplest analogy is the eye color. You see, we all have genes that encodes the color of our eyes, but some of us have blue eyes and some of us have black eyes.
I think I might be now, buddy.
Wow!
And in this case, gene that encodes blue eyes we call blue allele, and gene that encodes black eyes black allele. So we all have gene that encodes eye color, but this gene can be different, and different variations which are blue and black colors of the same gene that in this case encodes eyes we call alleles. So in this case, we all have gene that encodes MHC II receptor. We call this gene HLA-DRB, but this gene can be different, and different variations of HLA-DRB gene we call HLA-DRB alleles. For example, if person has HLA-DRB1 allele, this person will have MHC II receptor that was made based on genetic information in HLA-DRB1 allele. And such MHC II receptor presents substance in a perfectly normal way. And when T helpers income to this receptor, they recognize this substance as normal, and because of this, they do not see any danger.
And you can call me Papa.
If person has HLA-DRB3 allele, this person will have MHC II receptor that was made based on genetic information in HLA-DRB3 allele, and such MHC II receptor presents substance also in a perfectly normal way. Thereby, there will be no inflammatory response. If person has HLA-DRB7 allele, this person will have MHC II receptor that was made based on genetic information in HLA-DRB7 allele, and such MHC II receptor also presents substance in a normal way, so there will be no inflammatory response.
I have a son now. Little half-squat.
Who in crocky fuck is half-squat?
He's my son.
He's my boy. He needs me. I have to go back to him.
But some HLA genes do not know how to make MHC II receptor that will be able to present substance to T helpers in a normal way. So let's suppose that HLA-DRB4 allele encodes the last MHC II receptor. So in this case, person will have MHC II receptor that was made based on genetic information in HLA-DRB4 allele. The problem is that this particular MHC II receptor do not know how to present exactly this substance to T helper. So instead of presenting this guy in a normal way, they are doing this.
I think we were supposed to be doing this.
We did.
And instead, we did this.
And now, when T helpers income to MHC II receptor, they look at this guy and they think, who the hell are you? Get out.
Alright, but right now, you're like that kid on the playground, you know, the one who has lice that none of the other kids want to play with.
So because in this case, substance is presented in a normal way, T helpers cannot recognize it, and thereby they think that it's antigen. In response to any antigen, T helpers immediately becomes activated, and subsequently they begin autoimmune reaction against substance.
I was wrong! Blow the bridge! Blow the fucking bridge!
So in this case, we can say that person with HLA-DRB4 allele has genetic predisposition to autoimmune disorder, which is related exactly to this substance. So such person will be in a risk group for autoimmune disorder.
Great. Fine. Yep.
Ciao. What's that mean? Ciao. It's Italian. Means food.